Hi. In this video I'm going to talk us through a presentation we gave in 2019 on some foundational work we titled DeepPolyomics. This work looked to combine signals from MRI, blood transcriptomics and genomics into a single diagnostic factor that could help predict prostate cancer and determine if prostate cancer was likely to be aggressive. So before we dive into the findings in this work and why it's so important, I'd like to share a few terms that are relevant to understanding the work in this presentation. Now, polyomics is a term coined in this work to describe the combination of blood data, imaging data, and genetic data together. And what it represents is combining this information into a single signal that we can then interpret to determine the likelihood that a given patient, in this case, has prostate cancer. The next term relevant to this work is the idea of MRI Invisible prostate cancer. MRI Invisible in this case means that when a human observer reads the MRI scan, they are unable to see the tumour in the scan. This can be because some tumours do not express as readily in MRI and are invisible to the human reader. And finally, Gleason Grade. So there's two different types of Gleason Grade. There's a grading score and the grading group. But in both cases, it's an assessment about different grades of prostate cancer and their severity. Grades above 6 in the old system or groups 2 and above in the new system are typically considered clinically significant and worth considering primary intervention on. So let's get into the work. What we found in this publication was that from previous work we knew we could take genetic data about biopsy tissues and separate patients into 6 unique clusters, one of which in this work and previous work titled Cluster 5 was known to have more likely recurrent cancer and more aggressive cancer. In this work we first showed that on the 2 independent data sets we were looking at, those with additional information like blood data and radiology data, these same clusters were present when looking at the genetic data. This C5 cluster continued to show the lowest rates of recurrence-free months, meaning that the disease came back faster and often in more patients. So once we'd shown that, we started to look at this idea of polyomics. Now, polyomics in this case was looking at a combination of genetic data, transcriptomic data from the blood, and radiomic data from the MRI scan. So we extracted a number of features from MRI scans using a well-known radiomic pipeline, which takes the image data, extracts certain metrics and measures, things like textures and various values that can then be used as numerical features. It takes a complicated image and boils it down into a small group of numbers. Similarly, on the transcriptomic data, which is from the blood, you can boil down that data into a small number of numerical features. And this meant that for each patient that had genetic data, we had PSA values, age, radiomic, transcriptomic, and radiotranscriptomic data. We had a lot of numbers on this patient that we could start to use to train machine learning models. We show here in figures A through E that none of the metrics on their own showed good separation of the different Gleason grade groups. They weren't sufficient, that is, to determine who's likely to have prostate cancer, who's likely not to have prostate cancer, and in this case, what Gleason group they're in, which is how severe that might be. Figure F here shows a visualization about what the AI was doing in this case. It took a number of healthy patients and looked at the possible values for all of these different numerical extractions from the data, and when a new patient was compared, they were put into that same 3D space in this visualization, and based on how close they were to that known range of healthy values, we could determine whether they were likely to have prostate cancer and ultimately what the Gleason group was likely to be. So we use this to tackle a couple of problems. One, being this idea of MRI invisible tumors. So interestingly, in this work, we found that the Polyomics model could segregate MRI invisible and MRI visible tumors. Now, what this means is quite interesting. It's not only that we were able to predict who was likely to have cancer, despite them being invisible in MRI, but some combination of the genomic factors and the radiomic factors were actually able to predict who's likely to have a tumor that is in fact MRI invisible. Now, why this is so important and why this reflects on the work we do at Maxwell Plus is that we know that it's not just one data source that's going to get us to a diagnosis. And combining this very complex data with technologies like machine learning allows us to unveil hidden information within that data that can help us get patients to an early and accurate diagnosis. This was then independently verified, predicting the Gleason grades on the Cancer Imaging Archive data. We were there able to successfully separate patients into their Gleason groups and be able to predict who was likely to have high-grade or low-grade prostate cancer. So overall, what this means is that this model and this preliminary work that we demonstrated was able to nicely stratify patients into low and high risk categories, pick out previously invisible MRI tumors, and really set the foundation for the work that's come since, which is a number of AI models coming together to provide the patient ultimately with an opportunity for an early and accurate diagnosis.